Introduction. The Krebs cycle, also known as the citric acid cycle or the tricarboxylic acid cycle, is a central metabolic pathway found in the cell. It plays a crucial role in generating energy in the form of ATP and is intricately connected to various other metabolic pathways. Understanding the mechanisms, regulation, and implications of the Krebs cycle is essential for medical professionals, as dysregulation in this cycle can have significant implications in human diseases. In this script, we will delve into the various aspects of the Krebs cycle, discussing its overview, the enzymes and intermediates involved, energy generation, regulation, metabolic adaptations, interactions with other pathways, and its links to human diseases and therapeutic targets. Let us now explore each of these topics in detail. The Krebs cycle is a series of chemical reactions that occurs in the mitochondria, the powerhouse of the cell. It acts as a hub for numerous metabolic pathways, playing a central role in cellular respiration. The cycle begins with the entry of acetyl-CoA, derived from various fuel sources such as glucose, fatty acids, and amino acids. The cycle consists of a series of enzyme-catalyzed reactions, which ultimately result in the generation of ATP, reducing equivalents in the form of NAD and FOD2, and intermediates required for biosynthetic pathways. Through these reactions, the Krebs cycle effectively extracts energy from the fuel molecules, facilitating cellular processes and sustaining life. Energy production is a fundamental function of the Krebs cycle. During the cycle, energy-rich electrons are harvested from the fuel molecules and transferred to electron carriers, NAD+, nicotinamide adenine dinucleotide, and FAD, flavin adenine dinucleotide, forming NAD and FOD2, respectively. These reducing equivalents are vital for ATP synthesis. The electrons from NAD and FOD2 are subsequently transferred to the electron transport chain, a series of protein complexes embedded in the mitochondrial inner membrane. This electron transfer drives the synthesis of ATP through oxidative phosphorylation. Thus, the Krebs cycle serves as a crucial link between energy-yielding reactions and ATP synthesis. The Krebs cycle is a complex pathway involving several enzymes and intermediates. The cycle begins with the condensation of acetyl-CoA and oxaloacetate, catalyzed by the enzyme citrate synthase. This reaction forms citrate, the first intermediate in the cycle. Subsequently, citrate undergoes a series of enzymatic reactions, including isomerization, oxidative decarboxylation, and oxidative phosphorylation. Notable enzymes involved in these reactions include aconitase, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase complex, succinyl-CoA synthetase, succinate dehydrogenase, fumarase, and malate dehydrogenase. The intermediates, such as citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, and malate, serve as substrates or products of these enzymes, maintaining the flow of the cycle. Before entering the Krebs cycle, pyruvate, produced through glycolysis, undergoes a series of reactions to convert it into acetyl-CoA. This conversion occurs in the mitochondria, where pyruvate is first decarboxylated by the enzyme pyruvate dehydrogenase complex, resulting in the formation of acetyl-CoA. This decarboxylation reaction releases carbon dioxide and facilitates the transfer of electrons from pyruvate to NAD+, forming NAD. The acetyl-CoA thus formed can readily enter the Krebs cycle to initiate the energy-generating reactions. The entry of acetyl-CoA into the Krebs cycle is regulated and tightly linked to the energy demands of the cell. The levels of key intermediates, such as citrate, alpha-ketoglutarate, and NAD, act as feedback regulators to control the rate of acetyl-CoA entry. High concentrations of citrate and NAD inhibit the enzyme citrate synthase and alpha-ketoglutarate dehydrogenase, respectively, slowing down the entry of acetyl-CoA. Conversely, low levels of these intermediates and increased cellular energy needs stimulate the activity of these enzymes, promoting acetyl-CoA entry. This regulation ensures that the Krebs cycle operates at an optimal rate, efficiently producing energy as required by the cell. Decarboxylation reactions play a vital role in the Krebs cycle, resulting in the release of carbon dioxide and the generation of reducing equivalents. Multiple decarboxylation reactions occur during the conversion of isocitrate to alpha-ketoglutarate to succinyl-CoA, and malate to oxaloacetate. These reactions are catalyzed by the enzymes isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase complex, and malate dehydrogenase, respectively. Decarboxylation contributes to the energy generation process and enables the regeneration of key intermediates required for the continuity of the cycle. The Krebs cycle is a significant source of reducing equivalents, primarily in the form of NAD and FOD2. Multiple reactions within the cycle involve the transfer of electrons to electron carriers. For instance, during the conversion of isocitrate to alpha-ketoglutarate, NAD+ accepts a pair of electrons, resulting in the formation of NAD. Similarly, during the conversion of succinate to fumarate, fat accepts electrons, producing FOD2. These reducing equivalents are crucial in subsequent oxidative phosphorylation reactions, contributing to the synthesis of ATP. The Krebs cycle is tightly regulated to maintain metabolic homeostasis and respond to cellular needs. Several factors influence its regulation, including substrate availability, allosteric regulation, covalent modification of enzymes, and hormonal control. Key regulatory enzymes, such as isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase complex, are regulated through feedback inhibition by ATP, NAD, and succinyl-CoA. Additionally, hormones, such as insulin and glucagon, play a role in modulating the activity of critical Krebs cycle enzymes to adapt to nutrient availability. Anaplerotic reactions are essential in replenishing the intermediates of the Krebs cycle. As the cycle consumes intermediates for energy production and biosynthetic processes, these reactions ensure the maintenance of cycle integrity. For example, pyruvate carboxylase catalyzes the carboxylation of pyruvate to form oxaloacetate, thereby providing a vital intermediate to initiate the Krebs cycle. Other anaplerotic reactions involve the conversion of certain amino acids and organic acids into Krebs cycle intermediates, contributing to the overall metabolic flexibility of the cell. The Krebs cycle exhibits remarkable adaptability in response to nutrient availability and the energy demands of the cell. It can efficiently switch between different fuel sources, such as glucose, fatty acids, and amino acids, depending on their availability. During periods of fasting or increased energy needs, the cycle can be fueled by amino acids or fatty acids, while in the fed state, glucose becomes the primary substrate. This metabolic flexibility allows the Krebs cycle to coordinate with other metabolic pathways, ensuring the energy demands of the cell are met. The Krebs cycle is tightly interconnected with various other metabolic pathways, ensuring the efficient utilization of resources and the maintenance of metabolic balance. For instance, the Krebs cycle interacts with glycolysis through the conversion of pyruvate to acetyl-CoA. Additionally, anaplerotic reactions and the glyoxylate cycle provide links between the Krebs cycle and amino acid metabolism. These interactions enable the efficient exchange of metabolites and facilitate the utilization of substrates across multiple pathways. Dysregulation of the Krebs cycle is implicated in several human diseases. Genetic mutations or dysfunction in the enzymes involved in the Krebs cycle can disrupt energy production and metabolism, leading to disorders such as mitochondrial diseases and metabolic syndromes. Targeting the Krebs cycle and its associated pathways has emerged as a potential therapeutic strategy for combating these diseases. Modulating key enzymes, intermediates, or regulating factors of the cycle offers promising avenues for drug development and therapeutic interventions. The Krebs cycle, as a central metabolic pathway, performs a crucial role in energy generation, utilizing diverse substrates to produce ATP and reducing equivalents. It is regulated intricately, ensuring optimal function and adaptation to nutrient availability and cellular demands. The cycle's interactions with other metabolic pathways and dysregulation have significant implications in various human diseases. Understanding the complexity and importance of the Krebs cycle provides medical professionals with critical knowledge to diagnose, manage, and explore therapeutic interventions for associated disorders.